Okay, we are going to discuss the eukaryotic cell cycle. The eukaryotic cell cycle is basically the life cycle of the cell. There's going to be 5 main phases. The first phase is known as G1. The second phase is known as your S phase. The third phase is going to be known as your G2 phase. The fourth phase is known as your M phase. And the fifth phase is known as your cytokinesis phase. Now, each one of these actually stand for something different. Now, the first 3 actually are a sub-phase of a cell cycle that we call interphase. When we talk about interphase, interphase is the longest cell cycle. So these 3 phases basically make up a phase of the cell which we know as interphase. Interphase is the longest portion of the cell cycle. So I like to draw a circle to describe this. So if you say that the cell cycle starts, it's a cycle, which means it's going to go in a circle. It starts here. Most of your cell life is going to fall within interphase. So this is going to be broken down into 3 phases. So interphase has your G1 phase, which we're going to discuss. It's going to have your S phase, which is second. And then the third phase of interphase is called G2. These are all phases of interphase. It's the longest of a life cycle. 3/4 of the cell spends its time within interphase. Now, to understand the different phases of interphase, G1 is known as gap phase 1. This is going to be a time of cell growth. Okay. That is the very first thing that happens. So this is where it starts. In G1, this is the first thing that happens. So you start here, and it's working in this direction, the cell life cycle. In the first phase, it grows. The second phase is known as the synthesis phase. The word synthesis means to make. This is whenever you have DNA replication. DNA replication is when you make a copy of your DNA. So you take your chromosome and you're going to make sister chromatids. So we start with growth. This is phase 1. Then you go to the S phase and you're going to replicate your DNA. The second phase is called, the third phase is called gap phase 2. During gap phase 2, your chromosomes are going to condense and become visible. And your organelles, such as your centrosomes, are going to duplicate. Now, the reason we duplicate our organelles, centrosomes, is so that we can actually divide our DNA. So, in the previous video, we talked about eukaryotic cell cycles. In the S phase, you're going to take your linear chromosome and you're going to duplicate them. and you're going to make sister chromatids that are going to be held together by the protein's cohesion. That all happens in the S phase. But in phase 2, your chromosomes condense, but we need to divide the organelles, centrosomes, to duplicate so that we can actually divide the DNA that we duplicated. So the next phase is known as mitosis. Mitosis, let me change pens, is literally the division of the genetic material within the nucleus. Now, mitosis is divided into 5 phases. All of mitosis is going to have 5 different phases. So, mitosis, which we see here, has 5 phases. The 5 phases, I always say, are P-MAT or P-MAT. Now the dash represents one of the phases. So you have your prophase, which is always first. You have a pro-metaphase, which is second, that's the dash. Metaphase. Anaphase. And telophase. These are the phases in order. P-MAT. That's how you remember it. So, that's the end of phase 2. So, interphase. Then you have mitosis. That's when you divide the genetic material within the nucleus. Well, the last thing that you need to do is cytokinesis. And then the cell is going to start over. So I'm just going to put a C right here. When you talk about the term cytokinesis, the word cytokinesis means the division of cytoplasm and the cell finishes dividing into 2 cells. So we're going to have this life cycle, and when it divides into 2 cells, then that new cell starts its own life cycle. So this is the easy way to remember the phases. G1 is growth. Synthesis means to make, so we're going to replicate our DNA. G2, we need to duplicate the centrosomes. If you remember what the centrosomes look like, they're the microtubule organization center of the cell. You need 2 of these to be made so that you can actually divide your nuclear material. So you need to divide or make 2 organelles, the centrosomes. Mitosis is when you actually divide your sister chromatids, which were duplicated in the S phase. This is where these are going to divide. And then cytokinesis is where the cytoplasm is going to divide.